Hello, I'm your host, Ray Dogum, and welcome to VibeCast. Thank you for joining us as we explore the exciting advancements in technology-enabled collaboration to excel in important drug development. Vibe Bio seeks to find every cure for every community. We think big as no one should be left behind in the pursuit of living a healthy, happy, and productive life free from disease. VibeCast is our weekly informational podcast where we explore some of the hottest topics in drug development and technology innovation with some of the dynamic people that make up the Vibe community. Join us to learn, imagine, question, and help us identify and develop solutions together. Our guests today are Fabrice Chouraqui, CEO partner of Flagship Pioneering and CEO of Cellarity, and Laurens Kruidenier, Chief Scientific Officer. Cellarity is actually based in Somerville, Massachusetts, which is local to us here in Boston. So I'm super excited to speak with you both today about Cellarity and your experience in drug development. Welcome. Maybe we can get started, Fabrice, with you and just talking to us about your experience so far and what led you here.
Thank you, Ray. I'm very happy to be with you today. So I'm Fabrice Chouraqui. I'm a CEO partner at Flagship Pioneering and the CEO of Cellarity. I am like many of you who are listening us today. I am someone who is absolutely passionate about progressing medical sciences and bringing innovation to patients. I've always seen our industry as a kind of bridge between science on one end and society on the other. And I truly believe that the progress of science, technology, computation is really revolutionizing the way we are discovering and developing drugs. And we are just at the eve of a new era in therapeutic research. And that's really what brought me to Flagship Pioneering and to Cellarity. I really wanted to be acquainted and experience firsthand what technology can offer in unleashing, I would say, the power of the science and unlocking treatments that are out of reach today for patients.
Ray, super nice to meet you. My name is Laurens Kruidenier. I'm Chief Scientific Officer here at Cellarity. Joined about a year and a half ago. For all my career, I've been very passionate about scientific discovery. Really started early in my career focusing on immunology, particularly in the intestine. So diseases like Crohn's disease and ulcerative colitis have been focused on for a long time. And did that in academic research as well as in larger pharma companies before ending up in biotech. And I think biotech is really an exciting place, like Fabrice mentioned, where I think we can bring true innovation to patients. Previously, I was CSO at a company called Prometheus Biosciences in San Diego, where we actually developed the first precision medicine approach for patients with inflammatory bowel disease, and joined Cellarity because I think, and it will hopefully become clear in the podcast today, because at Cellarity we really are looking for new ways to discover drugs in ways that have not been possible before.
Excellent. I appreciate that. And it's cool. I actually worked in a lab for a few years working on intestinal stem cell stuff.
Did.
So
okay. Excellent.
extracted many crypts and stem cells from.
Yeah, I know how hard that is.
Yeah, we modified that protocol dozens of times. So let's get started with some questions, and this can go to either of you or both of you. What's been the actual traditional way drug companies have sought to discover new medicines so far? And what are some of the limits to that approach?
Actually, the process to discover novel medicines has hardly changed in the past decades, despite the evolution of science and technology. I mean, we know that diseases are complex biologically, and we've been using, in a sense, a reductionist approach to find simple key to unlock treatments. So to do so, we've been reducing complex disease to a single molecular target, and we've been placing a bet on a specific target very early in the drug discovery process. Sometimes it works. Unfortunately, most often it doesn't, because human biology is extremely complex. The pursuit of a treatment in NASH is a good example. I mean, empirical studies have suggested a plethora of targets to address the NASH disease. And actually, over the past 5 years, there's been a number of trials which have been studying a very large number of targets. And yet today, in 2023, there is not a single FDA-approved treatment in this disease.
Which disease? Was that NASH disease? Can you just...
NASH.
Okay. And what does that stand? I'm not too familiar with NASH. Maybe
It's a chronic non-alcoholic induced liver disorder.
Yes.
Yeah.
Okay.
Yeah.
I appreciate that.
I think maybe to add a little bit to Fabrice is saying here, I think those limitations of really reducing complex disease down to a single molecular target, I think were partly also technology driven. We're now in a time where technologies are evolving very, very fast. And this is both like analysis technologies, like single cell technologies that we use, and we'll talk a little bit about that more. But I think also computational approaches, right? You hear a lot in this field about AI, machine learning. We now are actually at for the first time, I would say, in history, actually able computationally to analyze and understand large amounts of data that we can generate with these new technologies. And to me, that's very compelling, because now actually we don't need to have to be in this place where we have to reduce disease complexity down to that single target. We can actually, if you like, embrace complexity and really appreciate, I would say, the complexity that the disease biology offers us, and therefore open up, I think, new ways of thinking about disease biology, and therefore new ways of actually treating those diseases.
Yeah, and that makes a lot of sense. Like the traditional way of using or identifying molecular pathways and finding targets for that specifically, I think made sense before. Now that we have thinking more like holistically, the complexity of disease in general and the number of variables that you'll have to be able to track, measure, and predict in a way makes so much sense for AI, right? Artificial intelligence can do this. Humans can't really do this in an efficient way. So I think it totally makes sense taking this approach.
I think it's a great point, Ray. The other, I think, point that humans are often is biased, right? We have certain preconceptions. I think, at least that's how I was trained as a scientist, is very much like reductionist, right? Reduce your problem as soon as you can to something simple that you think you understand. And I think that very quickly introduces bias. I think the beauty, also how we operate at Cellarity, very like an interdisciplinary manner where, you know, we have traditional scientists, biologists working together with computational biologists and scientists, to me also allows us, or actually gives us a new opportunity to take that bias out and let the data actually drive where we go rather than what we think we know.
Right. It's interesting, professors maybe that have been in the space for 30 years sort of like probably have like a hard time letting that go, that way of thinking go.
Right.
So there's a lot of change in the industry happening now as well.
Actually, for me, that was one of the compelling reasons to join Cellarity, and about something I enjoy every day, sort of the unlearning, right? It's forget what you know, let the data tell you what you actually know.
So Cellarity, having, this company that's focused on cellular level drug discovery, what's some of the major use cases for doing that?
I mean, so as I was saying, we clearly acknowledge the shortcoming of the current target-based drug discovery process, although this process has allowed to actually uncover amazing treatments. Yet it is quite inefficient, and that's why we believe it's time for a new approach. It's time for an approach that relies on a much fuller understanding of disease biology. And that's why at Cellarity, we've decided to fundamentally redesign the way drugs are created. And as the name of the company tells you, Cellarity, we focus on the cell. And focusing on the cell has a major advantage. As Laurens alluded to, by focusing on the cell, we are able to see biological system in full and not through the proxy of a single target or a single pathway. And by shifting the focus from a single target to the underlying cellular dysfunction, we are able to unravel the complexity of disease biology to create medicines that are, in a sense, out of reach with the traditional target-based drug discovery approach. And that's very unique, and that's very much at the core of what we are doing.
So can you share an example of how this approach has led to the identification of, a potentially new therapy, and maybe specifically what cell is being looked at or engineered?
Yeah, I think great question, Ray. Thank you. I think a good example is our sickle cell disease program. So sickle cell disease, as many listeners may know, is a genetic disorder. It's driven by certain variants in the adult hemoglobin gene. And the impact of that is that the hemoglobin is not fully functional. It basically polymerizes, the red blood cells that contain the hemoglobin turn from these floppy fluid cells into rigid sickle-shaped cells, hence the name of the disease. And downstream, that can cause a lot of problems for patients, which is can be very, very severe. Actually, the life expectancy of patients is only 40 to 50 years old. So it's a very severe disorder. The silver lining here is that we have another form of hemoglobin. It's called fetal hemoglobin. It's a version of hemoglobin that's expressed when you're a baby, when you're still in utero. And the beauty of that is that it's driven by different genes. So the mutations have no impact on that version of fetal hemoglobin. So this offers approaches, I think, especially for Cellarity, because like I mentioned, this disease is driven by red blood cells. So it's really a cell-driven kind of disorder. And what it allowed us to do is to ask the question is, what if we can find drugs that basically convinces these early red blood cells to not produce adult hemoglobin, but actually instead to produce fetal hemoglobin? Because then whether a patient has these mutations or not doesn't make any impact. These mutations will no longer have any impact. And we know from clinical studies that this works. So there are gene therapies currently in development, right, that actually try to correct the genetic variant. It's cumbersome, it's expensive, it's actually very risky treatment for patients. So if we can actually create a small molecule that would basically drive that same kind of effect, producing the fetal hemoglobin, I think that would be a massive game changer. So how we approached this was basically to deploy our platform. We took red blood cells from adults, we took red blood cells from fetuses, essentially. We profiled them at the single cell level. And that allowed us to understand what pathways we would need to activate to actually move these early cells from cells that produce adult hemoglobin, which we don't want, to cells that produce fetal hemoglobin. And we were able to quickly identify compounds actually that had a very impressive impact on that. And the beauty of our platform as well is that we can immediately compare the impact of our compounds with the impact that standard of care has. There are some small molecules out there that are not ideal, that do a similar kind of thing, but not in a fantastic manner. And actually, we can also compare this with how gene therapy works, what the impact of gene therapy is. And the beauty of our approach was that very quickly, we were able to identify compounds that drove this fetal hemoglobin production in a very significant manner that actually was superior to standard of care and on par with gene therapy. So I'm super excited about this because this actually allows us to say, potentially we have something in our hands here that's like a gene therapy-like efficacy, but now in a convenient small oral molecule pill package, right? So I think that would be an enormous value for this particular patient population.
I'm sure the audience actually must ask themselves, how are we doing what we're doing, given what Laurens said. So if you want, Ray, I have a small animation that I can use actually to say a bit more about how our platform works.
Sure. Yeah, I love that.
Let me pull it.
Absolutely. Thank you.
So as Laurens said, we start with health and disease tissue, and we collect transcriptomic profile of all the cells in that tissue. And these input allow us to build what we call a Cellarity map, which you see building here, which is, in a sense, a digital representation of transcriptomic data and other cell behavior data at the level of the single cell. So as you look at these maps, each columns represent a specific transcriptomic profile, and the height of the column tells you how many cells in that particular tissue has the same specific profile. The columns are arranged based on the similarity to each other. So the map reveals the cells that make up a tissue, diversity of their behavior, and the relationship between them. At this stage, we then compare a Cellarity map of a healthy tissue with a Cellarity map of a diseased tissue to really identify the cellular dysfunction between health and disease. And this comparison let us identify the network of genes that are changing between health and disease. So our goal here now is, in a sense, to solve for the cellular dysfunction. And we do this using what we call our intervention library. The intervention library is a proprietary Cellarity asset that we have built to link pharmacological intervention with the change in gene network that they induce in different cell contexts. So if I know the gene network that are changing, when cell move from a state of health to a state of disease, then I can use our intervention library and deep learning model to search our library to predict drugs that will address that change. And in the example that you see on that animation, our algorithms are predicting 3 different interventions that can address the cellular dysfunction that we have uncovered. And because our approach is target agnostic, because we don't start from a target, we are able to identify compounds with different molecular structure, which acts through different pathways on different targets. So obviously, we validate those predicted compounds in vitro and in vivo. And we use now the insight provided by those various predicted compounds to engineer new chemical entities. We have developed also some very cool machine learning features that identify the critical elements of those successful interventions. And they do this by learning from the structure, the protein mechanism, the network effects of the intervention that we have predicted. So as you can see, really what we have developed is a platform that really provides a highly differentiated approach to discover drugs that really act at the level of the cells to really modify the disease.
That's incredible. Like, I think this is potentially so revolutionary. You're basically adding another dimension to the process of drug discovery here through, especially with your intervention library. I think that's really interesting. It seems like you'll be able to do years of work in a matter of minutes and seconds,
Right.
potentially, right, with this model. So does this...
To me, that's a really good point. I want to just drill on a little bit, if I may, Ray, because at least during my career as a sort of a discovery biologist, what I spent most of my time on over the last couple of decades is target discovery, right? You take a disease, you take healthy, and you start to figure out which particular protein you think is really driving the disease. It's a lot of difficult, hard biology work. And then the end of it, actually, it's just a hypothesis. You don't know if that particular target in the clinic, if you block it in humans, in disease, is actually going to have an impact. It's a huge bet. And between that target discovery and that outcome, that clinical outcome, you're going to spend tens and tens of millions of dollars, right? To basically allow you to answer that question. So it's inefficient. It's a big bet. It's a big gamble. And it can lead to a lot of frustration, not in the least, of course, with patients. So the beauty, I think, of our platform is that you don't need to start there, right? You actually can come to chemistry like the animation showed very quickly, right? You can start to think about treatment before you actually fully understand the biology that you're driving. And by actually finding that chemistry, you can use that chemistry actually to figure out the biology. I want to come back to the sickle cell disease program because we actually applied this process there, where we were able, through this approach, to very quickly find these drugs that actually drove that fetal hemoglobin production. And because we had multiple compounds doing it, and because we could learn from the similarities of these compounds, we actually discovered a new pathway. We discovered new biology that actually drove that fetal hemoglobin production, right? So we got to a new biology, we got to a new pathway, but not by spending years and years digging in the literature, doing experiments to find it, but because we were able to quickly go from these Cellarity maps through our chemistry and use the chemistry to actually identify the target. So it's a little bit of a topsy-turvy approach, but I think it's very efficient and very exciting, because I don't think personally that the target and the pathway we would have identified if we hadn't had this platform. And of course, that unlocks now, hopefully new treatment options for patients with this disorder.
I think Laurens is highlighting something very important. So clearly, the platform that we are building, which is operating at the confluence of machine learning, biology, chemistry, high-dimensional data, allows to go faster and identify novel biology. But it also allows to do what it's designed to do. It is to increase the chance of success. I think we don't realize enough that drug discovery is very inefficient still now. As we speak, only 9 out of 10 drugs that are entering clinical development will reach the patient. 9 out of 10. Imagine if in the construction industry we had to build 10 skyscrapers to ensure that only one would be livable. I mean, this is insane. And that's actually what we are experiencing in the industry.
And the
And I'm
cost.
not even actually talking about the discovery phase where actually there is a lot of attrition. I'm only even focusing on when the program is reaching clinical development. And obviously this cost of failure has a major impact on the cost of discovering and developing new medicines. That's why actually, I mean, according to the latest estimate, it costs about $2.5 to $3 billion to develop a drug. It's not only the direct cost, it's the direct cost plus the cost of failure. And because of this high cost, medicines actually are very often priced very high. And that limit obviously access. Only a fraction of the population across the world, but also even in the Western world, can access the latest innovation. And that's, in my opinion, it's a problem. I mean, why we do what we do, it's to ensure that we can discover treatment that will significantly enhance human life. So all patients, will be able to access those treatments at the right time in their disease. And so since the cell is a much fuller representation of disease than just a single molecular target, clearly the platform that we have designed is clearly developed to drive much higher clinical success. And that we hope it will change the game, as Laurens said, in terms of speed, in terms of finding new biology that we can drug, but also in delivering treatment which a much higher clinical success, and as a consequence decrease the price of the medicine that we uncover, and obviously significantly increase access as a consequence.
Yeah, I mean, it sounds like this innovation can really change the way we view drug discovery entirely. And you mentioned sickle cell disease as one area that you've been focusing on, affecting hemoglobin, red blood cells in particular. But does this approach also lend itself to any other types of disease, other types of cells particularly? And are there certain diseases that it's more difficult to do this with?
Well, of course, I don't think Fabrice and I would be sitting here if it only would work in sickle cell disease. So the answer obviously is yes. So the company was founded in a couple of years ago, and really, I think the first couple of years we've been road testing this platform. Right. So we've basically been looking how our platform performs against multiple different diseases, multiple different cell types. And I can tell you, we've seen success in almost all of those. We've worked on type 2 diabetes, we've worked on oncology, we've worked on lupus, we've worked on other lung diseases, liver diseases. We started to probe a little bit in brain diseases. All the time we start to really, in every case, I would say we found interesting new hypotheses, pathways that would allow us, I think, to look at these diseases in different manners. So that also poses a challenge as a small company, right? In a world where you can do anything, you cannot do everything, right? We just don't have the resources for that. So we have to make some choices. So like I mentioned earlier, hematology, I think, is a good starting point for us because many of those disorders are driven by a single cell type, right? So I think lens is really as well as a stepping up stone for our platform. Immunology, I mentioned, I'm an immunologist, I think is this next step up where we have maybe the same cell players like we see in hematology, similar cell players, but maybe interacting with each other in a little bit more complex manner. And then all the way to NASH, right? The example that Fabrice highlighted, where we have a solid tissue, difficult to access, multiple cells are probably playing a part in this disease. That's why we also think that this disease is so hard to tackle, because, a single isolated protein may not be the sole driver of that particular disorder, right? So it's more complex. So we're also interested in those. In the future, and I'm also really excited by this, it's not some area we're into yet. You can really, I think, take this platform anywhere. We've been toying a little bit with quote unquote diseases like aging, right? Which, again, it's unlikely in my mind that aging, for example, is caused by a single protein, right, in your body, and therefore a very specific drug would stop you from aging or make you younger again, right? So I think, and I'm laughing a little bit here because it's a little futuristic, but absolutely, I don't see why, we would not be able to compare, complex cell behaviors in the aging process and find, more complex drivers of that process. And with our intervention library, actually would be able to find molecules that could intervene in that process. I don't see any reason why not. So I think the sky is the limit. Today we need to focus. We want to get to the clinic, demonstrate that we can do what we do. But I think beyond that, I think you'll hear a lot more from us.
What is the reception like from big pharma companies in regards to this type of approach? Is it something they're also investigating and interested in, or is this something that they're sort of, I'll just leave it at that.
This pharma companies see our approach as very complementary to what they do already. Many pharma companies, many very large pharma companies actually came to us thinking that our approach could help them address some of their pipeline needs, through a differentiated approach. They already invest millions, billions of dollars every year in drug discovery, and they are very eager to have multi-shot on goals. And that's what the Cellarity platform can provide. So a unique, a novel and unique approach to uncover novel actionable biology and design non-intuitive drug candidates that they can then proceed to the clinic.
How do you envision the future of drug discovery from a societal perspective, in addition to the scientific perspective? We talked about the scientific perspective in some lens, but as a society, what does this mean to families and patients and people in general? What can they expect?
Our industry has a major societal impact. I mean, we've seen it during COVID time. I don't think we would probably be here talking if companies like Moderna would not have developed the mRNA technology that gave us a vaccine in a record time. So this is really top of mind in everything we do. That's why very regularly we invite patients to come to our offices. We engage with them. It gives us, the energy, the purpose to do what we do every day. It's not easy to progress the science. It's not easy to push the boundaries to the science. It's not easy to develop new technologies, to pioneer. But when you know the needs of the patients, when you know that you have something in your hands that potentially can help address those needs, it's very compelling. It's absolutely compelling. So that's what we are doing. We will not limit ourselves to just one disease area or one indication. We clearly aspire to using this radically new approach to discover medicines into many different areas, as Laurens explained.
Yeah, absolutely. And if you think about the work that scientists do, like yourselves, it's hard to stay inspired because you have 9 failures for every 1 success, if you're lucky, right? And then being able to stay motivated and keeping patients at the center sort of helps keep the engine going, so to speak. So I appreciate that very much. Yeah.
Well, that to me is really the crux of it, right? I think we basically want to create more products, right? I think to see, there are, I think on an annual basis, 50 FDA approvals, right? In any given year on average. 50 new products, for an industry that spends, billions and billions and billions of R&D dollars, it's just not enough, right? So I think, we're both driven, Fabrice and I, by I think that desire to bring, new products out there that patients will need. I think the other point to me is also about technology, and innovation. I think the creativity, right? To think, how can we use these new technologies, these new computational approaches in a manner that's really creative and creates these kind of new opportunities. To me, that keeps me going very much, and we all do this across the industry, right? You can think about the gene therapy is a good example. It's new technology. It brings something to the table. And I'd like to think like that, how can we bring something new to the table, but that's actually more accessible, right? Utilize technologies in a way that's going to result, in great societal benefit.
Right. So we talked a lot about the potential of this. What are some of the real challenges that you're most concerned with? Is it finding the data to enter into your library? Is it getting regulatory approval for certain things? What are the real challenges you're facing now?
I mean, you mentioned one, data. Clearly, when people think about AI or machine learning, they think about algorithm. But one need to realize that the algorithms are as good as the quality of the data set that we are able to produce, and they can run on. And so we've learned as over the past few years, as we've developed Cellarity, that we really needed to identify, integrate, curate very high quality data sets. First, to avoid bias. So the curation is absolutely important. But also, this was the absolutely critical baseline for what we are doing, and to allow our algorithm that we are developing to really generate the value that we expect. So data set is something important. So how are we doing? We are combining public data set that exists. Unfortunately, they are not necessarily of equal quality. And so we have developed a unique approach to be able to identify them, to QC them, to ingest them. And we are supplementing those public data set with data set that we are actually producing ourselves out of clinical samples. We have an amazing team, very knowledgeable in multiomics data sequencing generation. And just to give you an idea, for instance, just last year alone, we have sequenced more than 15 million cells, which is very, very significant, I would say, for a company. Data is a challenge. Perhaps at the opposite of the spectrum, I would say talent is also a challenge.
Right.
As you've hope you understood by now, as we are working at the confluence of different fields, we are developing a new generation of talent, who can be who needs to be acquainted with different fields. And it's not necessarily easy. Those people often speak different language. If you think about a computational biologist, that individual will often look at the world in a binary fashion. If you think about a biologist, I mean, for a biologist, an experiment is never right or wrong. It never totally fails, or it's never totally successful. There is a whole spectrum of outcome. So when you bring those two people together, one looking at the outcome of an experiment in a binary fashion, the other looking at it from a spectrum of possibilities, it's not necessarily easy for them to work. So we have been spending a lot of time actually to educate people so they can learn from each other's languages, mindset, so they can really work in a very integrated fashion. And in a humble way, this has been not easy, but clearly I think we've been able to create one of a kind of scientific community at Cellarity, who now works in a fully integrated way across those disciplines.
Yeah, I think it's actually, I've also been encouraged by sort of the newer generation of scientists. I think that we see, being interested in the work we do, right? I think there is a clear desire to be multilingual, if you like, right? I remember when I was studying biology back in the day, the thought that I would cycle to, a company or to a building where people were sitting behind computers, I don't think it ever crossed my mind even once, So my mind was completely blocked from that possibility. I think whereas now we have new generation of scientists that are actually very keen to collaborate, and we just need to help them do that, right? And I think to me, again, comes back to the unbiased, it comes back to creativity. when all these disciplines come together and have a shared goal like we have, I think you can create some real fireworks here. And we see that almost every day, which is really exciting to me.
That is very interesting. And, helping identify what skills they need in order to succeed is really, I think, the core challenge there. And once they figure that out and there are resources for that, it's all a matter of put the work in and kind of like
and focus, right? Focus on the goal.
Focus. That's another big issue in our society in general these days, with so many things taking our attention.
Right.
And to be fair, this has played to our advantages, and we've been able to attract actually amazing talents from the best universities, obviously in Boston. They are just across the street, or even across the world. I think people know that by joining Cellarity, they'll be exposed to cutting edge science. They'll be able to learn to do drug discovery in a different way. And obviously that learning, they'll be able to apply that learning going forward. And they can see that it is, in a sense, next generation drug discovery that they are doing. So that has really played in our advantage to be able to attract amazing talents, knowing that we are in competition with top companies, not only in the pharma sector, also for our machine learning specialists and computation folks. We are also competing with the best of the technology firms, often on the West Coast.
Switching gears a little bit, so I know Cellarity has raised a Series C in 2022, raised a bunch of money, which is great in order to continue the science. And maybe this question is more for Fabrice, because you've been with you're working with flagship pioneering for some time. So the question is, are you seeing any kind of innovation in the biotech funding landscape? How companies in biotech are actually funded?
As we see the integration of biology and technology, obviously, I believe that we will see really change in the funding environment. I mean, today it's fairly segmented. People invested in biotech with a capital B are often investors who are very knowledgeable in biology, drug discovery, drug development. With drug discovery operating now at the intersection of technology, biology, we see that tech investors are more and more interested in what we're doing. And I believe that we will see biotech with a capital B moving to biotech with a capital B and a capital T.
Yeah, that's a really interesting point. There are a lot of LPs, VCs who are interested in the biotech space, but don't have the understanding of all the complexities that have to do with biotech and investing in that space. So being able to find ways to communicate that science so that they can understand it more clearly and feel more comfortable investing in these potentially revolutionary technologies is really key to keep pushing forward here. So I appreciate that answer, and it makes a lot of sense. Fabrice, Laurens, I want to say thank you so much for joining me today on VibeCast. I really appreciate your time and sharing a little bit about Cellarity and your experience with us. So is there anything else you'd like to leave the audience with before we conclude today with our show?
Right. First of all, thank you so much for having us. I believe that we are on the cusp of a new era in drug discovery, in therapeutic research. It is a fantastic time, in my opinion, not only for scientists, but for doctors, caregivers, patients. I think we are like we've never been in the past. We are now able to provide new hopes in many different diseases. The next milestone will be obviously to make sure that this innovation can be accessed broadly across the world. And that is something that, as an industry, we have a duty to make it happen. And that's a whole other topic for another podcast, perhaps. But I think this is something that we should strive hard to doing.
Yeah, thank you very much, Ray. And it sounds like you'll need to have us back in a year or so, and we can tell you where we are then.
Absolutely, I'd love that. I appreciate that. Well, thanks again, guys. Talk to you soon.
Thank you.